Hello, my name is Associate Professor Andrew Geering. I work as a plant pathologist at University of Queensland within the Centre of Horticultural Sciences at QAAFI. Sunblotch is potentially a very serious problem. It can affect the yield of a tree by 80%, reduction by 80%, and it also disfigures the fruit as well. The fruit has very unsightly scars on it, so the fruit becomes unmarketable when it's affected by sunblotch. In Australia, we've had an active control campaign since the 80s, and for us, the main problem with avocado sunblotch is that it's a trade impediment. Other avocado growing countries want to prevent entry of the viroid into their country. When you export fresh fruit, it has viable seed in it. The seed can transmit the viroid at rates of 90 to 100%. So we want to provide evidence of freedom from the pest in orchards. So then the orchards or the farmers are then registered to export their fruit. Our dilemma, as opposed to, say, a vegetable grower, is our commodity is a tree that's 10 metres tall by 5 metres wide, and there might be a population of 20,000 trees in an orchard. And the question is, how can we efficiently sample the orchard, being such a large area and such large trees? The viroid, when it infects the tree, it infects every part of the tree, including the pollen. The bees are brought in to pollinate the trees as a normal farming practice to improve yields, and they collect the pollen and they take it back to a central location and parcel it in a little cell within the beehive. So they've done the sampling work for us. So all we have to do is open up the hive and sample the pollen and test that, rather than a team of people climbing up ladders or going onto cherry pickers to collect leaves. Well, I think it's a very versatile technique. The bees are reliable and they're free and they're very thorough in their sampling activity. They sample all parts of the tree. They also can detect any virus or pathogen in the tree that's represented in the pollen can be found. So we can test for a suite of 10 or 20 different plant pathogens at once. Well, next we would, I guess, like to roll out the technique to a region. So, for example, the Atherton tableland is now the largest avocado producing area in Australia, rivaled by southwest Western Australia. So we'd like to intensively look at beehives around the Atherton tableland, and now we've demonstrated the principle, then roll it out to a region and see if we can demonstrate that the Atherton tableland is free of the pathogen, and then that would lead to the region being declared pest-free, and there wouldn't be any barriers to exports of fruit.